again want to welcome everybody to our HERN webinar series. Most of you have come to these types of webinars before. This is a series put on by HERN. Maybe a few of you may not know what HERN is. It's an NIDDK funded network. It's got two major goals, really. First, to address beta cell loss in type 1 diabetes and try to figure out ways to protect or replace beta cell mass. And the way that NIH decided to do this was to create a network. And again, most of you are familiar with the network, but what they did was they created different themes to look at different aspects of in the field. And so by creating this, it allowed and brought together a lot of different people around the world to collaborate with one another. You can find out more about HERN and more about this network on our website. But essentially, we've got over 200 different investigators, many different institutions spread across at least 9 different countries. Again, I did mention the website. It's for you here. And one of the things that we're doing is we're cataloging a lot of the resources, some of which you might hear today in Decio's talk. You can go to our resource browser, and there it's again for you on the right-hand side, on the upper right-hand side, where there is many different things. Insulin antibodies, for example, different mice that might be of interest to you, protocols, and you've heard some of these talks before, all of which are cataloged on our YouTube channel. So today's talk as well will be made available. So if you want to revisit that, it'll be posted here. All right, so without further ado, this is the agenda that Decio promises to bring to life for us. And I won't go over those in detail. He will, but I want to remind you, please try to keep yourselves muted so we can really minimize the disruptions during the talk. But afterwards, hopefully we'll have plenty of time for a Q&A, and we're going to try to reserve a large amount of time, in which case I'll invite you to talk, and please unmute yourself and we can have a really good discussion. So while I switch, and I'm going to stop sharing and Decio will share, I just wanted to say a little bit about him. I think most of us know Decio. I've been really excited to hear this talk, and I think most of you would agree with me since this was announced. Decio is just a bona fide beta cell biologist. I think at heart, he probably is also a beta cell immunologist as well. He's published over 500 papers. Some of those papers have even been cited over 1,000 times or close to it. It's hard to sum up all of his work, from ER stress to inflammation to gene regulation to alternative splicing, lots of different areas, but it's going to be a real pleasure to hear about what he has prepared for us today. So without further ado, Decio, I turn it over to you and look forward to your presentation.
Thank you very much. It's a pleasure for me to speak to you today, and I have been member of the HERN since the beginning, and I think this is a fantastic initiative. So I will just start by reminding you about a philosopher who lived in the 12th century, Moses ben Maimon, and as many philosophers in this period, he was also a physician. And one of his major works was the Guide for the Perplexed. And if he would be alive today, it's conceivable that he would address one question that perplexes many of us: why the immune system kills beta but not alpha cells in type 1 diabetes mellitus. And the data I'm going to show is the product of the work of many people. You have the ones who really participated in the work are indicated here in bold font. And I will come back to the names as the lecture go on. This is the financial support, including, as you can see, the NIDDK and HERN. And the human samples that we used They were provided by NPOD, the human islets by Piero Marchetti, and a lot of the histology that I'm showing on human islets were prepared by Sarah Richardson and Noel Morgan. And we collaborate with people all over the world, as you see here, including both immunologists and islet biologists. And again, I will come back to these colleagues as the lecture proceeds. So this is a human islets of Langerhans that you all know well. And you see that in the human islets of Langerhans, the alpha cells in red are in close proximity to the beta cells. However, when you have, let's say, insulitis, inflammation, and eventually the induction of type 1 diabetes mellitus, you'll have the invasion of the islets by macrophages, and perhaps the most important killers here are CD8 T cells, who will kill the pancreatic beta cells. And the killing will take place by cell-to-cell contact and also by release of pro-inflammatory cytokines. One thing that is puzzling, if you look at this picture in one of our papers, it was done by Sarah Richardson and Noel Morgan, we were looking here at the PD-L1, but pay attention in the glucagon positive and insulin positive in a normal glycemic individual. And if you go to see, you see the islet of a patient with type 1 diabetes of long duration. You see that all insulin-producing cells disappeared, but the alpha cells remain. So you can have a situation here where all beta cells have been killed while the alpha cells remain. This does not mean that the alpha cells are physiologically sound. So there were some quite interesting papers by Marcela Brissov and colleagues, basically showing that glucagon secretion is defective in alpha cells from patients affected by type 1. And Klaus Kestner was kind enough to share with me a paper that he, Doris Stoffer, and other colleagues have accepted in Journal of Clinical Investigation, which basically showed that alpha cell function is abnormal in individuals who are GAD positive. So these are normal glycemic people who at the very early stages of what may become type 1, and their alpha cells are already dysfunctional. And why are these alpha cells in type 1 not working normally? One possibility, as Marcela Brissov and colleagues suggested, was that the extreme beta cell loss and systemic hyperglycemia contributes to the alpha cell dysfunction. One thing to keep in mind, at least in the first years of disease, is that the alpha cell dysfunction is often characterized by hypersecretion of glucagon, while beta cells will be producing less and less insulin. We were curious of what's happening to the alpha cells, and what we decided to do together with Piero Marchetti and Emanuele Bossi and Guy Rutter, we downloaded the HPAP single cell databases, and Emanuele matched type 1 donors and controls, and did the same with type 2 donors and with 5 matched controls. And the idea was to test two following possibilities. One, that the alpha cell dysfunction would be disease specific, and in this case, we would see different genetic signatures in type 1 and in type 2. The other possibility being that the alpha cell dysfunction was perhaps a consequence of the beta cell failure, and then we would see similar gene signatures in both cases. As you can see here, there is very little similarity in the upregulated and the downregulated genes. when we compare alpha cells from type 1 and type 2 patients. And indeed, when we did the pathway analysis of these alpha cells in type 1 and in type 2, you see that in type 1, the alpha cells present at the top signatures related to immune disease, while in the case of type 2, it seems that the signatures are not immune mediated. So the conclusion that we can have, this is a preliminary conclusion, is that the mechanism of alpha cell dysfunction is different in type 1 and in type 2. And it's conceivable that in type 1, this dysfunction is secondary to the autoimmune process. However, I remind you that these alpha cells, they are dysfunctional, but they are alive. So even after 20, 30, 40 years of disease, these alpha cells remain. So the question is how it comes that the beta cells are killed while the alpha cells survive. So let me go back a little bit of some preliminary data that will lead us to the main subject of today. So we have been very interested in the role of type 1 interferons in type 1 diabetes mellitus. So you see here the signal interferon alpha will bind with surface receptors. It will induce JAK1 and TYK2. This will lead to phosphorylation of STAT1, STAT2, and will lead to an antiviral state and inflammation. And together with Carmela Evans-Molina and colleagues, we are studying TYK2 inhibitors as a potential protective agent in type 1. Interferon alpha is probably important because it is present in the islets of patients with type 1, and it has been implicated in the early stages of the disease. And one thing that Laura Marocchi in our lab together with Reynald and other colleagues showed is that interferon alpha induce inflammation, HLA class 1 and ER stress in pancreatic beta cells, and these are three hallmarks of type 1. And the other thing that is important is that polymorphism in TYK2, and now it's clear also in JAK1, are candidate genes for type 1. So we were curious, and we have been studying the let's say, gene and proteomic signatures induced by interferon alpha in adult human islets of Langerhans. And when you compare, for instance, the gene signatures induced by interferon alpha in human islets against the gene signature observed in islets from patients with type 1, and this is a paper by Russell and colleagues, and our comparison was published a couple of years ago, you see that there is a lot of similarity in the upregulated genes, suggesting that indeed patients with type 1 diabetes have an interferon alpha signature at the level of the islets. One possibility is that this is just secondary to islet stress, but this is not the case because when we compared islets from patients with type 2 against human islets treated with interferon alpha, there was very little similarity. However, I started to see, because in many patients, the autoimmunity against beta cells will start very early in life. It's conceivable that even intrautero or in the first months of life. And we started to wonder if it wouldn't be worthwhile to also characterize the impact of type 1 interferons on immature human beta cells. And in our lab, together with Miriam Knopp and colleagues, we have established an IPS facility where we can differentiate inducible pluripotent cells up to, let's say, a immature human beta cell, and which would be useful for this purpose. And these cells, they have a nice insulin and glucagon expression, and we have shown a couple of years ago that already at this stage they express receptors for interferon alpha, interferon gamma, interleukin-1 beta, and tumor necrosis factor. So what we did is to use this inducible pluripotent derived islet-like cells for RNA sequencing. So before doing RNA sequencing, we characterized the cells, so they have around 50% beta cells, around 10 to 15 glucagon positive cells. They already respond to glucose, but it's a miserable response. It's around a two-fold increase compared to 5 to 6-fold in an adult human islets of Langerhans. But importantly, they are susceptible to interferon alpha, which, as we do in adult, as we see in adult human islets of Langerhans, induce HLA class 1, the chemokine CXL10, and the protective protein PDL1. So our idea was to do single-cell RNAseq on these preparations. However, we need to keep in mind something that was very nicely shown in a review by Mark Housing and colleagues, is that single-cell RNA sequencing is very useful when you want to discriminate gene expression in particular cell types, but with good luck, you will detect 5,000 genes. So basically, you will pick the highest expressed genes. While when we do bulk RNA sequencing, and we usually do a deep sequencing, we can detect more than 100,000 variants, including alternative splicing. I am not going to discuss alternative splicing today, but it will be the subject of a talk at a symposium at the ADA this year. So as a compromise, we decided to do in parallel bulk RNA sequencing and single-cell RNAseq. So if you look first at the bulk, it's very similar to what we see in adult human islets of Langerhans. So you see all the usual suspects, all the HLA-related molecules. And when you look at the pathways, let's say induced by interferon alpha, it's also very similar to the adult human islets of Langerhans. So you get, of course, JAK-STAT signaling, you get the type 1, you get interferon signaling, antiviral responses, and so on. And when you look at the inhibited genes, you see, for instance, inhibition of oxidative phosphorylation, the respiratory electron transport, and so on. However, things started to get more interesting when we went to the single-cell RNAseq. And this is the bioinformatics is done by a very talented bioinformatician in our lab, Florian Simzer, who is just submitting this paper. So when you start to look here, you see, for instance, that antigen processing cross-presentation seems to be higher in beta-like cells than in alpha-like cells, while the antiviral mechanism seems to be a little bit less intense in beta-like cells than in alpha-like cells. However, here we have put a cutoff, let's say in the log P10 p-value adjusted on 14. So what we did next was to put the cutoff instead of 14 at a much higher level and focus on the beta and alpha cell. And when we do this, we start to see some interesting differences. So for instance, interferon signaling, it's much more intense in the alpha than in the beta cells. Antigen processing, it's rather similar, but the antiviral mechanisms are again much more intense in the alpha cells than in the beta cells. We went a little bit further, and one of the things that we have shown some years ago is that more than 60%, actually 80% of the candidate genes for type 1 are present in human islets of Langerhans. And we started to look at the expression of these candidate genes in beta and alpha-like cells, and they are different. So we have been proposing, and I think now it's becoming popular, that we should also study candidate genes not only in the immune system, but also at the target cell level. And we often studied wall islets. And I think it's now becoming clear that perhaps we need to look at these candidate genes at alpha and beta cells in a separate way. But I mean, this is still ongoing work, and I will come back to you on a later occasion. So let's look now at some of the key RNAs that are modified by interferon alpha in beta-like and alpha-like cells. So this, what you see here is single-cell RNAseq. The size of this circle gives an idea of the percent of the cells that express this gene. So this big circle here suggests that nearly 100% of the cells express this gene. And the color indicates the level of expression. So a very bright red circle indicates, for instance, in this case, HLA-E, a gene that is present in most alpha and beta cells, and the expression is a little higher and in the alpha cells. So let's go a little bit further. The first thing is to make it sure that we are indeed looking at beta and alpha cells, and as expected, the beta-like cells express PDX1, while the alpha-like cells have a preponderance of expression of ARX, which is a classical transcription factor from alpha cells. If we go a little bit further, so as I told you before, alpha cells induce endoplasmic reticulum stress in wall human islets of Langerhans. However, when you look here at SHOP and XBP1, you see that the ER stress markers are much higher in beta than in alpha cells. So it seems that beta cells are suffering more from ER stress than alpha cells. And I'm going to come back to this later. The other thing that we look at are genes involved in protection. So BCL2L1 is the popular BCL-XL, and it is much higher in alpha cells than in beta cells. You look at HLA-E, which is an atypical HLA molecule, which has an inhibitory role on the immune response, and again, it's much higher in alpha than in beta cells. The same for BIP, which is very lowly expressed at basal level, and then it goes dramatically up when alpha cells are exposed to interferon alpha, while beta cells show the opposite phenomenon. They have a slightly higher basal BIP, they fail to respond to interferon. And then when we look at the HLA molecules involved in antigen presentation, they are more or less similar, perhaps slightly higher in alpha than in beta cells. So let's start to look, let's say, what's the potential functional impact of these changes. One of the things that is important when you are doing this type of analysis is to go back to histology of human islets of Langerhans in type 1. And here we are very lucky to collaborate with Sarah Richardson and Noel Morgan and with NPOD to get access to this precious material. So you see that, for instance, HLA-E, this protective molecule that seems to be much higher in alpha cells than in beta cells exposed to type 1 interferon. They are also, if you look in the pancreas of a non-diabetic individual, you see no HLA-E. When you have islets from people with type 1 who still retain beta cells, you see that there is a massive increase in HLA class 1 expression. And funny enough, this is a little bit difficult to see here, but expression is much more marked in alpha than in beta cells, which is in line with our RNAseq data. Let me just, I recovered from a non-COVID cold and my voice is not great. So let's take a look. This is a review that Roberto Malone and myself wrote some time ago, where we're looking at the weakness in the biology of the beta cell that may promote their autoimmune vulnerability. And let's compare, let's say, beta and alpha cells here. So the first weakness is that making insulin and other granule proteins promotes beta cell stress. Well, alpha cells are also doing a lot of glucagon, so this shouldn't be so different. However, and we're going to come back to this, alpha cell seems to be more resistant to endoplasmic reticulum stress. Islets are highly vascularized, which provides easy access for immune cells, and this should be the same for alpha and beta cells. And then in the early type 1, you have an upregulation of HLA class 1 processing, which, as we saw, is similar. However, alpha cells have a much higher expression of HLA-E, which is an atypical and protective one. And the third one, weakness 3, beta cells secrete their products in the bloodstream, which is similar for the alpha cells. So the first thing that we looked at was the resistance or vulnerability of beta and alpha cells to Coxsackie virus. And why this? Because there has been a lot of papers, and I am citing one here from Sarah Richardson and colleagues, indicating that a large proportion, but not all, of the beta cells in patients affected by type 1, they express VP1, which is a protein characteristic of Coxsackie virus. So we were curious to see how beta cells and alpha cells would respond when they were infected with Coxsackie virus B5. At that stage, we were able, this is, I'm speaking of 2012 to 2015, and this is the work of Laura Marrock in our lab, we couldn't as yet purify human alpha beta cells, so this was done initially in rat alpha and beta cells. So we are looking here at purified alpha and beta cells that were infected with Coxsackie virus B5, and we are following expression of VP1. So you see that VP1 goes up in beta cells, and by 24 hours they are falling apart, while it goes up in the alpha cell, and then it goes down. And if you look at this insert here, you see that there is a clear increase in VP1 in alpha cells, and then the alpha cells somehow kick the virus out. Why this? One possibility is that STAT1 and MX1, it's an antiviral protein, are much higher in alpha than in beta cells. And we were curious if this would be due to the fact that alpha cells have less expression of the viral receptor, and this was not the case. And then what we did was to infect these two cell types with an adenoviral vector encoding green fluorescence protein. This adenoviral vector enters using the same receptor as the Coxsackie virus, and you see that the number of positive cells is similar in beta cells and alpha cells. However, the average fluorescence is much lower in the alpha cells, which explain for people that work with alpha cells that these cells are famous for being difficult to transfect. And they are difficult to transfect because they are able to neutralize and then kick out the infecting virus. And Sarah and Noel, they had some samples from a neonate that unfortunately died with cardiomyopathy caused by Coxsackie, and this child has a massive infection. And when they look at the islets of this child, they could see that Coxsackie virus, VP1, was also present in the alpha cells. And we then infected dispersed human islets in vitro, and we looked at VP1 expression early. And if you look here, you could see that not only beta cells, but some of the alpha cells were positive. Well, this is an interesting story. So I am suggesting that alpha cells are more resistant to viral infection, in this case Coxsackie, than beta cells. Is this specific for alpha and beta cells? The answer is no. So there were 1 year, let's say, before our study, a very interesting paper in Nature Medicine, where they were looking at the brain of people who unfortunately died with Nile fever. And what they have seen in these individuals is that the virus induced a massive damage in the cortex, while the cerebellum is relatively protected. So they treated, in this case, cortex cells or cerebellum cells with interferon beta, and they looked at the genes. And what do they see is that under basal condition, STAT1 is higher in cerebellum than in cortex, as it is several of the genes that we have seen. So what we did was to pick the list of genes that were higher in cerebellum than in cortex and see how this compared, let's say comparing alpha against beta cells. And you see that there was a lot of similarity. So many of the genes that were higher in cerebellum compared to cortex, they were also higher in alpha cells compared to beta cells. And when we evaluated this in a systematic way, so you have here gene expression in the cortex, in the brain cortex compared to the cerebellum, and here alpha against beta cells, there is a correlation. It's not great, but it is significant. However, when we look at the number of genes, you see that you have 44 genes that are in common, that are overexpressed in cerebellum compared to cortex, and the same in alpha cells compared to beta. Well, I am saying that this is probably related to a vigorous innate response immunity, which is regulated by STAT1. To test this hypothesis, Laura knocked down STAT1 in the alpha cells, and when you knock down STAT1, so you can see a nice knockdown here, you see that the antiviral responses like MX1 go down, and when you do this, the alpha cells become susceptible. So when you knock down STAT1, the Coxsackie virus now is able to establish itself and proliferate in the alpha cells. This is rat data, and we are trying to reproduce this in human alpha cells, which is not a trivial work. So this is one part. The other thing that I mentioned to you is that alpha cells seem to have a more vigorous resistance, let's say, to endoplasmic reticulum stress. I think most of you are familiar with endoplasmic reticulum stress, so I don't need to explain it again. But let's see how alpha and beta cells respond to endoplasmic reticulum stress. And to do this, we went to type 2 diabetes mellitus, which also induces a vigorous endoplasmic reticulum stress. And this is data done together with Miriam Knopp and Piero Marchetti. So Piero and colleagues, they did electron microscopy of beta and alpha cells from 7 controls and 7 individuals affected by type 2. And this was also published by Laura Marrocchi and Piero, Miriam, myself, and other colleagues. And one of the ways to evaluate endoplasmic reticulum stress by electron microscopy is to look at the surface volume of the ER. So for instance, if you look at the beta cells in the non-diabetic abetic islet donors, the value is 0.9. In the type 2 donors, it goes up more than threefold. So this is one of the mechanisms of adaptation to ER stress. There is an increase in size of the ER stress. If you look at the alpha cells, their endoplasmic reticulum stress also increases in size, more or less to the same extent as the beta cells. However, when you look at cell death, you see that there is an important increase in cell death for the beta cells, while the alpha cells remain smiling. So there is no mortality in the alpha cells. We were puzzled by this, so we decided to expose human islets of Langerhans in vitro to palmitate, which is a well-known metabolic stress to the beta cells. And when we look at the ER of the cells exposed to palmitate, both in beta cells and in alpha cells, there is a clear-cut increase in the endoplasmic reticulum area, which is pretty similar. However, you see beta cell death, and again, the alpha cells are smiling. So palmitate is inducing ER stress in both beta cells and alpha cells, but while beta cells die, the alpha cells remain alive. Why this? And here we went back to the rat model. One of the possible reasons is that, as we have seen in the single-cell RNAseq, BCL2 and particularly BCL-XL are much higher in alpha cells than in beta cells. This was done in rat, but our single-cell RNAseq indicate that this is also true for human alpha cells compared to beta cells. Is BCL-XL, which is a key anti-death protein in the alpha and beta cells, really relevant here? Probably yes, because again, Laura knocked it down, in this case, BCL-XL, and you can see that there is a very nice knockdown, let's say comparing the small interfering BCL-XL against the small interfering control. And when you knock down BCL-XL, the protection that alpha cells had against ER stress is lost. So now alpha cells become sensitive to endoplasmic reticulum stress, and you have a quite important level of alpha cell death. So if we go back to this nice figure, actually it was done by Roberto Maloney, the original figure, I just tempered it with it a little bit here. What I try to do in this figure would be to add, let's say, what's different between alpha cells and beta cells. So the first point is that making hormones may promote beta cell stress while alpha cells fare better. And why this? I think one of the reasons is that alpha cells are much more resistant to endoplasmic reticulum stress than beta cells. And this is true for rat beta cells, and it's probably also true for it's true for rat alpha cells, and it's probably also true for human alpha cells. If we look to inflammation, alpha cells are better, they can better endure viral infections and pro-apoptotic signaling than beta cells. And this is again probably related to ER stress, but also probably related to BCL-XL. And then when we look at the HLA class 1 processing pathway, which seems to be similar in alpha and beta cells, however, it seems that the immune system does not recognize alpha cell antigens. We don't know why, and this is something that we are looking together with Roberto Maloney. One of the reasons may be due to the fact that alpha cells has much higher levels of HLA-E, which is an inhibitor, atypical HLA molecule, than beta cells. So if we put all this together and go into free speculation mood, what can we I wouldn't dare to say conclude. I would prefer to say suggest. Well, one thing that we can say for sure is that both pancreatic beta and alpha cells suffer in type 1, but while beta cells are killed, alpha cells survive. Exposure of IPS-derived islet cells to interferon alpha, which is most probably an early cytokine in type 1, induces both similar, e.g., HLA-related genes, and different, for instance, BCL, XL, ER stress-related defenses, innate immune response, and antiviral genes when comparing beta-like and alpha-like cells. One thing that is important also is that expression of candidate genes for type 1 diabetes also shows several differences between beta and alpha cells. And now going to the speculations, it seems that the capacity for antigen presentation is similar between beta and alpha cells, but either alpha cells are somehow less antigenic than beta cells, perhaps due to increased HLA-E, or their capacity to endure viral infections and ER stress allows them to better survive viral and or immune-mediated stress, leading to less alpha cell death and consequently less presentation of antigens to the immune system. These are hypotheses that remain to be tested. I think this is what I would like to share with you today, and I will be very happy to listen to your views and answer questions if I can.
Thank you very much, Decio. We really appreciate the presentation. What I'd like to ask you all to do is, would you please raise your hand, just given the number of people we have, I want to try to do this in an orderly way. Go down to the bottom of the screen, push the reactions button, and you can raise your hand and ask our speaker a question. And so we'll call on the order that it is presented. Hi, Sasenka, please go ahead and unmute yourself and ask Decio your question.
It's a great talk. I've got two small questions for you. First one is, we showed that, ER stress induces alternate splicing of BCLX that favors the BCLXS variant, which is apoptotic. Are you suggesting in alpha cells that there's something with the alternate splicing mechanism that favors the XL variant to be formed?
We cannot say this because, as I told you, when we do single-cell RNAseq, we just detect 5,000 genes. So we cannot go down to splice variants. So what we are going to do is bulk RNAseq of purified human alpha and beta cells. And once we have this data, which may take a couple of years, we may be able to answer this question, which is indeed an important question.
The other quick question I have is that to think about it the other way, are there conditions that kill specifically alpha cells and leave beta cells alone?
That's a very good question. I have never found it. There are some conditions that kill both of them equally. But something that kills preferentially alpha and not beta, I have never seen. So alpha cell seems to be hard cookies or hard nuts to crack. So I have never seen this. If any of you have seen situations where alpha cells are more susceptible, this raises an interesting evolutionary question. So why beta cells are more sensitive than alpha cells, even considering that beta cells are surely physiologically more important than alpha cells? And I think this is an open question. Perhaps
Thank you.
In 5 years' time for another webinar.
Thank you.
Thank you, Sasenka. Bart, please go ahead and unmute yourself.
Yep. This what? Can you hear me?
Yep. Go ahead,
Yeah.
Bart.
So this was great, Decio, as always. Just a small comment on another difference between alpha and beta cells, in addition to subtle differences in HLA-E, is of course the antigens, and particularly the neoantigens that they might express. I suspect that there might be also differences in ribosomal infidelity, for example, that might be worth to look into. But my real question relates to your iPS-derived beta cells, and you were very careful to call them beta cell-like. My experience is that they are less sensitive to perturbations than pancreas-derived beta cells. And I wonder whether you did the comparison with bona fide pancreas-derived beta cells to confirm that?
We are doing this now, but when we look at the global gene expression, it's pretty similar. And we published a couple of years ago a paper where we looked at cytokine-induced cell death, and they were rather similar to let's say, adult human islets. But clearly, I agree with you, these cells are not fully mature. But I think what I am suggesting here is that we can transform a lemon into a lemonade. Since autoimmunity probably starts very early, I think it makes sense to, besides looking at adult human islets of Langerhans, to also look at immature islet-like cells and see how they respond to an early cytokine. So this
no,
is
I completely agree, actually, Decio. I think it's that's why this is this has an additional merit, your work, to maybe show that if you take beta cells that do not make 500,000 or 1 million insulin molecules per minute, that you might be better off in terms of resisting, different kinds of stress. So I think this work serves multiple purposes, and I'm very thankful for you sharing this.
Let's hope that the referees of the paper agree.
Cheers.
Thanks, Bart. Sally, please go ahead and unmute yourself.
Hi, Decio. Thank you for a wonderful seminar. You said that alpha cells under stress are dysfunctional, but could you comment on the level of dysfunction upon stimulation of alpha cells that are under stress, any kind of stress?
This is actually not our work. So I am basing here the data that I presented is the work of Marcela Brissova. And
Okay.
now there is this very nice paper coming out. I think I saw Doris. If Doris is online, would you care to comment? Because you are the last author of this nice paper on alpha cell dysfunction in GAD-positive individuals.
Hi, Decio. Hi, everybody. I'm sorry, I missed the question. I was talking to my babysitter. So what was the question exactly?
The question was, what is the degree of alpha cell dysfunction, both in, let's say, individuals who are just GAD-positive and in people who already have type 1? And I think you have data to answer.
Yeah, so we, as you mentioned, and thank you for mentioning the work, we studied single antibody GAD positive. So most of the single antibody individuals are GAD positive. So we studied islets from 9 donors and compared them functionally to, I think, 6 type 1 donors and a pretty large group of controls. And what we saw was a notable partial loss of glucose suppression of glucagon secretion. And we also saw a potentiation of IBMX stimulated glucagon secretion, which is not seen in type 1. So in the type 1, we did study the function of that subset of type 1 islets, and we saw what had been seen in Marcela's paper, and even and also by us in previous work, a more dramatic loss of suppression by glucose, but not the potentiation of IBMX secretion. So that's sort of something we're thinking about for the future, whether that reflects something intrinsic to the alpha cell, which is suggested by the transcriptomic analysis we did, where we saw a pretty substantial downregulation of PKIB, which is a potent inhibitor of PKA signaling, or whether that reflects something altered about the paracrine environment. I think it's another interesting possibility.
Another thing that is interesting in your paper is how early this phenomenon is taking place. And the other thing, it's And I think we have seen this in beta cells, that dysfunction and death can be dissociated, and which suggests that perhaps the mechanism of dysfunction and death are not necessarily the same, which is an intriguing thought, because in general, intuitively you think, okay, you have dysfunction, and as the cells cannot take anymore, they will die. But this may be to some extent true, but it may be that there are also separate pathways, as we see in the alpha cells. This is something for future work.
Thank you. Thank you. I hope that answered your question, Sally. Ed, you're next.
Great. Thanks for taking my question. And thanks for taking the time. Great talk, everyone. So my question pertains to beta cell antigens that could also be expressed in alpha cells. So for instance, we know that GAD65 is expressed in alpha cells. And so one thing I noticed, Decio, is you seem to be insinuating that maybe direct cytotoxic effector cell killing is not necessarily what's killing beta cells. But if you did have a CD8 T cell, say for instance, specific to GAD, would it not kill alpha cells, even though they have upregulation of HLA and also express the antigen? And why would that be the case?
Well, apparently they don't kill. I am not insinuating that CD8 are not the main. I think this is well established by work of Bart Rupp and other people. I think it's by now clear-cut that CD8 T cells are the main inducers of cell death. Why they don't kill the alpha cells is, who, as you very well pointed, also express GAD. It's really a very interesting question. One possibility is this overexpression of HLA-E that alpha cells have. We couldn't detect PD-L1 in the single cell RNA-seq because the expression is not sufficiently high, but this is something that we are also looking. In the histology that Sarah Richardson and Noel Morgan did, PD-L1 seemed to be expressed in both, actually even more in beta than in alpha cells. So why the CD8 T cells don't kill the alpha cells, it's a mystery. It may be HLA-E, it may be because they have more BCL-XL who will block the intrinsic apoptotic pathway. It's funny because also we are doing with our IPS, and I think Bart and other people are also doing co-culture between CD8 and IPS-derived islet cells or adult. And it will be interesting to see what happened to the alpha and the beta cells if they are killed to an equal extent or if the beta cells go first. So I think that's a very interesting question. Thank you very much.
Thank you.
Thanks, Ed. Mazhar, please go ahead and unmute yourself.
Thank you so much. This was a great talk, Decio. I think one question I had was, you have shown a lot of data about alpha cell apoptosis in times of stress. I was wondering if there was any data that suggested that the proliferation signals of alpha cells was also increased. As we know that type 1 diabetic pancreas also have increased alpha cell mass, and if there was an increase in the proliferation signals as well.
We didn't pick it. I think this is an interesting question again for Doris and Klaus, because they are doing single cell RNA-seq of the real sink, which is pancreas from type 1 patients and controls. In the interferon alpha treated islet-like cells, we didn't pick any impact on proliferation. Doris, just calling your help again, did you see any modification in proliferation in the alpha cells?
No, we did not. We did not measure, like we didn't assess Ki-67, but certainly at the transcriptomic level, there weren't there wasn't anything pointing in that direction. There was also no, there was a lot of variation amongst the donors, but on average, no change in the alpha cell proportions. both by solution cytoph within the islet and also looking at, a surrogate of mass in the imaging mass cytometry, where we use the total pancreatic area as the denominator.
Thank you, Doris. Sharon, please go ahead and unmute yourself.
Okay, thank you very much. Very nice presentation. I just a question regarding the whole pancreas infection. Recently, we can see some data showing the virus infection is not just excluded to the islet, and we can see infection in the whole pancreas and mostly in the exocrine. I just want to know, do you have any data or idea to show these endocrine exocrine crosstalk leading to beta cell destruction?
We don't have any data in this direction, because I think this is better done in the actual pancreas from type 1 patients. And I think Klaus, Doris, and colleagues, they are also looking at the non-endocrine cells. Actually, I am calling so much Doris' help that next time that I have a lecture to give, I will make sure that Doris is there to help me to answer questions. But just coming back to your question, which I think is a relevant one, our model does not allow to answer it. I think this will need to be looked at in the single-cell RNAseq of the different pancreatic cells from patients with type 1. But it's interesting because, let me think a little bit, we are doing experiments with viral infection of IPS, but again, this IPS-derived islet-like, they are mostly endocrine cells, so we couldn't answer to your question.
Decio, I didn't think I would get a chance to ask my question, but I'm happy that it looks like I can. In those interferon alpha experiments where you were treating the alpha and beta-like cells, was there more genes that were similar across both cell types than there were differences?
That's a good question. We didn't check the number of genes. This is easily done. We will do a Venn diagram type of experiment and take a look at the genes. If this is an interesting question, we just didn't do it, but we can easily do.
Yeah, that certainly would be interesting to me. Decio, there was a lot of congratulatory remarks, very interesting discussion, comments in the chats, and lots of hand clapping. I don't see any further questions from anyone. So unless there's any last minute questions, yes, like Alejandro and others, I want to congratulate you, Decio, and thank you for putting together this very stimulating presentation. And hopefully we can have you again soon to give us some updates on some of these ideas and data that are just coming out. So yeah, I agree. I also thank you very much.
Thank you. Thank you for having me. It was a very interesting discussion and gave me some interesting ideas for additional experiments.
That's wonderful. Everyone, we'll hope to see you next month sometime. Look for the webinar announcements, and we'll certainly have another interesting discussion. Thank you very much. Bye-bye.
Thank you.
Bye-bye.
Okay. Bye.